How can you collect representative water samples for turbidity analysis? Imagine trying to get a clear picture of water quality, but the sample you take is not quite right. That can lead to misleading results, especially when measuring turbidity. So, how do you make sure your water sample truly reflects what's in the water? It starts with careful steps. First, use clean bottles made of plastic or glass. Make sure they are rinsed thoroughly with water that has no turbidity. This prevents dirt or particles from contaminating your sample. When collecting water from a natural source, avoid stirring up sediment from the bottom. Instead, dip the bottle just below the surface or at the depth you want to study. This way, you avoid adding extra particles that could skew the turbidity reading. Take enough water, about 250 milliliters or more, so you can run multiple tests or make dilutions if needed. After filling the bottle, gently swirl it to mix the sample without creating air bubbles. Air bubbles can scatter light and make turbidity seem higher than it really is. Wait a few moments for any bubbles to settle before testing. Temperature also matters. If possible, let the sample reach room temperature because cold or hot water can affect the turbidity measurement. When rinsing your bottles and equipment, use water that has no turbidity. For samples with very high turbidity, above 40 nephelometric turbidity units, you might need to dilute the sample with clear water. Measure the turbidity of the diluted sample, then multiply the result by the dilution factor to find the original turbidity. Be careful to avoid condensation on the bottles, as water droplets inside or outside can scatter light and cause inaccurate readings. Wipe the containers with a clean cloth before testing. Label each sample clearly with the date, time, and location. It's best to analyze the sample as soon as possible to prevent changes from settling or biological activity. Following these steps helps ensure your samples accurately reflected the true turbidity of the water. Proper sampling is key because turbidity can vary a lot across different spots and times in the same water body. Using a nephelometer or turbidity meter, which detects light scattered at 90 degrees by suspended particles, gives you the most accurate results. Remember, good sampling practices are essential for reliable water quality assessments and pollution tracking.